Hi learners, welcome to today's lecture on levels of protein structure. I am Dr. Gurunathan from Christo Jayanti College. To begin with, what is a protein structure? We know that a protein is basically a polymer of amino acids which are joined by peptide bonds, and there are 20 standard amino acids which are involved in it, and each of the amino acids in the protein requires a particular linkage between the amine group of one molecule and the carboxyl group of the adjacent molecule, which is called as a peptide bond. And this peptide bond is so important that the entire protein structure depends on the different peptide bonds that are formed between different amino acids in the polymer of the protein. And they have observed that if a polypeptide chain has a mass which is exceeding 10,000 units and the number of amino acids in the chain exceeds 100, it is usually referred as protein. Whereas the remaining molecules which contain 10 amino acids or more than 10 amino acids, which is very close to the 10 number, they together form a polypeptide chain. Moving down to the different levels of the class of protein structure, there are four levels of protein structure that are existing. The first level is called as the primary level or the primary structure of protein. The second level is called as the secondary structure of the protein. The third level is called as the tertiary structure of the protein. And the fourth level is called as the quaternary structure of the protein. Moving to the primary structure, it is the exact order of the amino acids which are present in the protein that is called as the primary structure of the molecule. And you should see that the change in the sequence present in this primary structure also affects the protein functionality. Not only functionality but also the structural aspect of the protein is also disturbed if there is any change in the sequence. And you see that many of the amino acids that are present in the primary structure, they are connected with each other because of covalent and the peptide bonds between the amino acids present in the primary structure of the molecule. Moving down to the next level of the structure in the protein, which is called as the secondary structure, they are formed because the local folds that may be observed due to interactions between the atoms of the backbone. Similarly, these folded interactions they actually are observed between the amine and the carboxyl group of the peptide link, and they have identified that two well-known structures of secondary elements are present. One is called as alpha helix, the other one is called as beta pleated sheets. And they have observed that the structure arises due to the regular folding of the backbone of the polypeptide chain, particularly the hydrogen bonding between the carboxyl group and the amino groups of the peptide bond. And they also observed that segments of protein chain may acquire an own local fold which is much simpler in its conformation and usually can take the shape of a spiral or an extended shape or a loop. Either of this can be there. These local folds are called as secondary elements and they form the protein's secondary structure. Among the two important secondary structures which are well known, first is the alpha helix, and this is one of the most common secondary structure observed, and it has a right-handed screw. And this is mainly formed because of the amino group of each amino acid residue and the carboxyl group, the adjacent turn of the helix, which is responsible for the formation of this right-handed screw-like alpha helix structure. Moving to the second structure, that is beta pleated sheet, which is a common secondary structure observed in protein molecules. We generally resemble the pleated folds of drapery. That is why it is called as beta pleated sheets. And you see that there are two forms of beta pleated sheets available in the protein structure. One is called as parallel beta pleated sheets, where both the lines of the say chains that are lying side by side are moving the same direction, whereas there is another form of beta pleated sheet which is called as anti-parallel beta pleated sheet, which is responsible for formation of the anti-parallel beta sheets based on the direction of the side by side chains which are existing between each other, and intermolecular hydrogen bonds that are existing between these side by side chains makes the beta braided sheets an interesting form of structure. Moving to the next level of your protein, which is called as the tertiary structure. And this level is because of further folding of the secondary structures of the protein molecule. And they are mainly stabilized by several bonds that are existing between the amino acids present in the molecule, such as hydrogen bonds, electrostatic forces, disulfide linkages, which are commonly seen between the cysteine residues, van der Waals forces, hydrophobic bonds between the hydrophobic residues that are present in the protein, and the ionic bond. Sometimes negatively and positively charged side chains will help in formation of the tertiary structure. And they have identified that this forms the 3D shape, which is also referred as the native conformation of protein molecule. In fact, they have identified that two major molecule shapes are formed in the tertiary level of the protein molecule. One is called as the fibrous molecule, the other one is called as the globular molecule. A typical example for a globular molecular structure contained in proteins is the immunoglobulins. Their tertiary structure gives a globular structure. That is why they are called as immunoglobulins. And this level is commonly okay for a single chain amino acids. Whereas when we move down to the next level, this is called as a quaternary structure. This is very commonly observed when you have more than one chain in the protein molecule. And these chains must have a proper spatial arrangement, and they should have a proper linkage between each of them so that the entire protein molecule now becomes functional. And a typical example is the one that everybody knows, that is called as human insulin, which is made of two chains, chain A and chain B. Both these chains are produced separately during the process of transcription and translation, whereas they undergo a proper linking between each other to finally form a functional insulin molecule. So this the overall picture is shown here, which begins with the primary structure, which is basically the amino acid sequence produced from the translation process, which is undergoing folding to form the secondary structures such as alpha helix and beta braided sheets. And these secondary structures undergo further folding, which lands up to form the tertiary structure of the molecule. And if you have more than one chain in the protein molecule, these chains interact with each other to form the quaternary structure based on the complex interaction of the protein chains present in the protein molecule. With this, I end up my session today. Thanks for your patient listening. We'll meet again another interesting video. Take care and bye.